Creativity is thinking up new things. Innovation is doing new things. Assalamualaikum warahmatullahi wabarakatuh. The International Diabetes Federation, or IDF, said that Indonesia has a diabetes alert status because it ranks 7 out of 10 countries with the highest number of diabetic patients. The prevalence of patients with diabetes in Indonesia reached 6.2%, which means that there are more than 10.8 million people suffering from diabetes per 2020. Diabetes mellitus type 1 is a metabolic disorder caused by an autoimmune reaction, causing a damage to pancreatic cell characterized by chronic hyperglycemia due to severe insulin deficiency. Blood sugar levels are controlled by the hormone insulin, which is produced by the pancreas. In diabetes patient, the pancreas is unable to produce insulin according to the body's need. Without insulin, the body cell cannot absorb and process the glucose into the energy. Type 1 diabetes is also known as an autoimmune diabetes. In most cases, type 1 diabetes mellitus also has more severe risks and symptoms than type 2. Therefore, the new innovations are needed in the treatment and healing step as well as the therapy for type 1 diabetes mellitus patients. From the origin of genetic engineering, there have been advanced technology that have sparked new enthusiasm and revolution with the latest CRISPR technology. Researchers can instantly change the DNA sequence of almost any organism, thus enabling the explanation of genetic function at the system level, as well as identifying the genetic causes of a condition or disease. Even a small change in a person's gene will fundamentally alter cell function. CRISPR technology has supported the researchers with tools like never before. CRISPR-Cas9 is known to be effective in gene alteration of the human pluripotent stem cell, or HPSCS. The process of altering the HPSCS gene associated with T1D is reduction of IPSGS from patients with T1D, triggering improved isogenous control and differentiation into the pancreatic cells. Key et al. 2015. The repair process of pancreatic cell can help the regulation of insulin in the body. Thus, it can help patients in treating type 1 diabetes. So regarding these problems, we, Lilis Nurfitriani, Siti Miftahul Jannah, and Kresna Purwandaru, with Mr. Muhammad Nurkolis, STP, MP, PhD, as our advisor from Brawijaya University, proudly present our innovation entitled CRISPR-Cas9, Cluster Regularly Interspersed Short Palindromic Repeat Associated Protein 9 Technology as a genetic therapy of type 1 diabetes mellitus patients.
There are three methods in writing this scientific paper. The first is the data collection stage. This study used data collection techniques random sampling through a questionnaire, where the questionnaire will be used to measure level of public interest in using CRISPR. The second stage of research was using qualitative research methods with data or information collection methods using survey methods through questionnaire and using library research methods that refer to several existing research journals. And the last is data analysis.
CRISPR associates protein abbreviated to Cas are endonuclease enzyme produced by the transform the Cas operon. CRISPR can change, repair, replace, delete, or insert part of DNA in cells and living things. The way CRISPR works is linked to the find, cut, and paste the system of a computer, where the search system will go through one part of the 3 billion base pair along the chain like Google search. The action of Cas9 depends on the presence of PAM or protein-splicing adjacent motif sequence on the target DNA so that it can recognize its own DNA or not. Furthermore, Cas9 will cleave unbind the DNA duplex and cleave both the chains, a double-strand break. make change and allow DNA to make natural repair through non-homologous enjoining. Repair pathway that are least accurate or an accurate homology-directed repair or ADER pathway.
Based on the research survey questionnaire that was distributed from 3 to 5 June 2021, we obtained as many as 86 respondents with an age distribution of 18 to 24 years. Based on the figure 1, it can be seen that there are 41.9% of respondents who still don't know the fact that Indonesia ranks 7 out of 20 countries with the highest number of diabetes patients. This shows the public knowledge and awareness regarding diabetes is still quite low. Even though diabetes is not a like problem, there are more than 10.8 million people suffering from diabetes each year 2020. The solution offered by the author is the development of gene editing technology and in Figure 2, as many as 80.2% of respondents think that the solution is right for overcoming the problem of diabetes mellitus type 1, while in Figure 3, as many as 97.7% of respondents agreed that the CRISPR-Cas9 innovation was implemented as a therapy for type 1 diabetes mellitus patients. After previously, respondents were given brief information about the description of CRISPR-Cas9. Then, as shown in the bar chart figure 4, it can be seen that there are 68.6% of respondents think that the CRISPR-Cas9 innovation is effective to be implemented as a therapy for type 1 diabetes mellitus patients. CRISPR-Cas9 can to be used in the long term as a therapy for patients with type 1 diabetes mellitus. Based on the data, it can be seen that the community has positive response to the innovation of CRISPR-Cas9 as a therapy for patients with type 1 diabetes mellitus.
And then, the diabetes mellitus is a metabolic disease consisting of type 1 diabetes mellitus caused by an autoimmune reaction of pancreatic cells and type 2 diabetes mellitus due to the body's inability to use insulin optimally. Type 1 diabetes mellitus is a disease that requires gene editing in this treatment. CRISPR-Cas9 is present in bacteria and archaea that play a role in adaptive immunity. The process of repairing pancreatic cells helps regulate the body insulin as well as treats type 1 diabetes mellitus. Based on the research and the questionnaire, the community has a positive response to the CRISPR-Cas9 innovation as a therapy for type 1 diabetes mellitus patients and so that this innovation has a great opportunity to be developed and implemented in long term.
That's all from us. Thank you so much for your attention. Stay happy, healthy, and hydrated.